Hello everyone, I'm Akanksha Singh and I'm Research Fellow at Pine Biotech, and I would be presenting my project titled In Silico Analysis of NGS Data of Glioblastoma to understand the effect of IDH1 status. I'm an undergraduate final year student studying BTech in Biotechnology at Amity University, Noida, India. My research interests are cancer biology, precision medicine, bioinformatics, and data science. Glioblastoma are type of brain cancer that are originated in glial cells and considered as most aggressive type of tumor. It generally ranked from grade 1 to grade 4 based on how aggressively they develop. Grade 1 glioma is gonna be least aggressive, whereas grade 4 glioma is also known as glioblastoma, gonna be very aggressive. Glioblastoma was originally characterized into two subtypes as primary glioblastoma and secondary glioblastoma. But according to classification done by WHO in 2016, where primary glioblastoma considered as IDH wild type glioblastoma and secondary glioblastoma as IDH mutant type glioblastoma. So what exactly IDH does? It is a metabolic enzyme occur in cytoplasm and peroxisome, which functions is to catalyze the oxidation of isocitrate to alpha ketoglutarate in citric acid cycle. It maintains redox balance and protects cells against oxidative damage. Whereas IDH1 mutation is a missense mutation lead to single amino acid substitution of arginine residue at codon 132 in exon 4, which result into accumulation of 2-hydroxyglutarate, which lead to conformational change at active sites. So for my study, I came across the dataset from NCBI GEO dataset as shown in the slide. There are 118 samples which consist of adult glioblastoma with 85 samples, neural stem cells with 15 samples, lower grade glioma with 18 samples, and on analyzing the metadata, I found that it has IDH status in only 86 samples. So, on the basis of literature that I reviewed, I came up with these points such as glioma patient with IDH1 mutation show better therapeutic responses and longer survival, which is remain unclear. Number 2, no novel therapeutic strategies have yet translated to clinical benefits based on IDH status. Based on these points, I designed my hypothesis as how gene expression vary in IDH wild type versus IDH mutant tumor. Number 2, biological pathways and respective function affected in glioblastoma patient with IDH1 wild type versus IDH1 mutant type. Number 3, looking for more differentially expressed gene in IDH status of glioblastoma that might be further targeted as novel therapeutics. So the methodology I followed for my analysis is as shown. The supplementary file that I obtained consisted of DSEC normalized data. So in order to analyze my data in a better way, I performed exploratory analysis first on all the data and then particularly in groups consisting of IDH1 wild type and IDH1 mutant type. Then I ran tsec2 pipeline to obtain significant genes. This was the pipeline I followed for my exploratory analysis. So this was the first scatter plot I got from analyzing all the groups consisting normal tissue, IDH1 wild type as well as IDH1 mutant type. But I did not get the clear overview of analysis, so performed another analysis. This was the second plot I came up with after analyzing two groups, particularly IDH1 wild type and IDH1 mutant type. But again, I was unable to get the clear idea about the clusters and the outliers. So I performed comparative analysis on two groups with the help of pipeline as shown. This pipeline consists of DSEC2 enrichment analysis and GSEA analysis. With the help of this analysis, I obtained information about significant differentially gene expressed, gene ontology, and KEGG pathways. This is the volcano plot, usually done under two conditions: the adjacent p-value at y-axis versus log2 fold chain on x-axis, which result in depicting significant differentially expressed genes. The blue area represents significant gene, whereas red area represents significant differentially expressed genes. Genes which are having negative log2 fold chain value will be considered as downregulated, whereas positive log2 fold chain value to be considered as upregulated genes. KEGG enriched pathway provides the information of enrichment of genes in the area displayed on the basis of two parameters, which are count and p-adjacent value. It is observed that the highest number of upregulated gene is associated with glutamatergic synapse and nicotine addiction pathway, while the highest count of downregulated gene is related with viral carcinogenesis pathway. This is gene ontology plot I got after performing DSEC pipeline. It is basically a ratio of genes that are correspondent in particular pathway to all differentially expressed genes on the basis of criteria of p-value. For example, there are approximately 200 genes which are involved in extracellular matrix organization. GO pathway or gene ontology pathway provides the glimpse about the function of genes which are highly activated due to upregulation of certain genes, as well as the function which are suppressed due to downregulation of some genes. In this network plot, we generally analyze the pathways based on fold chain value. We analyze the genes that are involved in particular pathway. In this plot, size of dot is the code depict the number of genes involved in the particular pathway, whereas color indicates the fold chain value. According to plot shown, CDCA4 and H2AC17 gene, which has the least value of fold chain and are prominently involved in chromatin organization, involved in regulation of transcript. Conclusion and discussion. This was the process that I was followed for selecting differentially expressed genes. My dataset has total 53,731 genes, but after running DSEC pipeline, I obtained 36,753 differentially expressed genes. Further, I filter p-adjacent value with less than 0.05 and I got 14,327 genes. And on applying further filter on log2 fold chain with more than 1.5 or less than minus 1.5, I left with 3063 significant differentially expressed genes. And I got the 1550 upregulated gene and 1513 downregulated gene from significant differentially expressed genes. Key findings are: from KEGG analysis, possible association can be made between suppressed pathway of viral carcinogen and neutrophil extracellular trap formation in glioblastoma. From geoanalysis, possible association can be made between activated pathway of immunoglobulin complex and suppressed pathway of platelet-derived growth factor and nuclear nucleosomes. Future directions. Much work is needed in future to assess the precise effect of IDH1 mutant pathway on progression of glioblastoma. Researcher can perform an experiments focusing on these particular genes found in the study and correlate their upregulated and downregulated gene with the disease progression. So this was the project I worked upon. Thank you so much.